Hi everyone. So this is the third part of chapter 3, and this is all about cells. And so you're probably familiar with this. You've probably learned about it before. So we'll go through all the parts of the cells and the organelles and what they do. And it's important because if you have a disease or disorder, sometimes it involves the organelles. So it's important to know how the cells work and what's in them. All right. So the cytoplasm is the first part, and we've learned that word before. That's just the kind of liquid goo, mostly water, that is in the middle of the cell, and in it has cytosol, which is that gel liquid. It's also got stuff in it like protein, salt, sugars. Then you've got inclusion, so you've got things that are not soluble in the cytosol, like little lipid droplets, the vacuoles that we've talked about, or vesicles and things like that. And then you have organelles, and these are special. You can think of them like mini organs, and they're special structures that the cell has. So here are some of the different organelles. Some are enclosed in membranes, so they're called membrane bound, and some don't have membranes around them. When we say membrane, we mean a phospholipid bilayer. So the ones that have membranes are mitochondria, the endoplasmic reticulum, which we'll call ER, the Golgi apparatus, peroxisomes, and lysosomes. They all have phospholipid bilayers around them. Ones that don't are ribosomes, cytoskeleton, and centrioles. Okay, so the first one's the mitochondria. We know it as the powerhouse of the cell. It's actually the site of oxidative phosphorylation, whereby ATP is made with oxygen, and that's called cell respiration. So the mitochondria does cell respiration. It has a double membrane around it, just like the phospholipid bilayer, and then it also has membrane inside that's the same. It's phospholipid bilayer. And it has all these little folds, and those are called cristae. And anytime you see lots of folds or wrinkles or layers in biology, that tells you that there's high surface area and that it's allowing lots of chemical reactions to happen. So these folds are called cristae, and this is where cell respiration happens. Mitochondria are also unique because they have their own DNA. They have different DNA from the rest of your cells, and you inherit your mitochondria from your mom. And so the DNA is actually similar to bacterial DNA, and so they think that mitochondria used to be bacteria. They're the same size, they replicate the same, they look the same, they have the same DNA, all of that. So they used to be bacteria that became engulfed by our cells. So here is a mitochondria. It has this little cristae in the middle, right? The little folds. Let me get my laser. And that shows us that there's chemical reactions happening there, and that's where the ATP is made from glucose. Okay, then we have ribosomes. These are super important. These are non-membrane bound. All cells have them, bacteria all the way up to us. They have in them, they are made of ribosomal RNA, which is made in the nucleolus, and they make protein. So the purpose of ribosomes is to produce proteins, to synthesize your protein. There's two types. Some of them are just free-floating in the cytoplasm. They're the free ribosomes, and then the membrane-bound ones are stuck to the ER, the endoplasmic reticulum, on the rough ER. And so both of those types make proteins. And so here's your two types of ER. Your ER is like a whole bunch of flattened, like wrinkled membrane, and it's made of membrane. They're not considered it's just phospholipid bilayer, just wrinkled together. And there's two types. There's the rough ER that has the ribosomes, and the smooth ER does not have ribosomes, and it makes lipids. So the rough ER makes proteins, the smooth ER makes lipids. So here is your nucleus, like the purple thing. Your nucleus has little holes in it for the mRNA to come out. And then right here is the rough ER, and you can see the little orange ribosomes on it. And then over here is the smooth ER, and there are no ribosomes on that. So you have rough and smooth. So the rough ER has all these ribosomes attached, and that's where protein is made. I love you, Mom. I love you. And it's also makes proteins. You know how the plasma membrane has little proteins embedded in it? That's where they are made too, and the phospholipids. And then they get processed later. So they're going to get enclosed into a vesicle, little membrane sac around them, and then they're going to go to the Golgi, and the Golgi is going to further process them. The smooth ER does not have ribosomes. It looks different from the rough ER, but it's like still little folds. They're just different looking. So this is where lipids are made, lipids are processed. It also absorbs, synthesizes, and transports fats. It detoxifies drugs, converts glycogen to free glucose. Glycogen is kind of like the starch in your muscles. And then it stores and releases calcium. So it does different things than the rough ER. Okay, then the Golgi. The Golgi is a kind of I think of it as like the UPS store of the cell. So the Golgi is going to take proteins that the ribosomes made, and it's going to modify them and package them into proteins. And it can transport them out of the cell. It can use them within the cell. It can put them in the membrane. They come in on the cis side of the Golgi, the inner side. It's called cis right here. Then they're modified, all of that sorted, packaged, and then they leave on the trans side, which is the outer side. So let's see. Make sure I'm on the right side here. Okay, I think this is this is the cis side. So it's coming from the ribosome, from the rough ER. This is the protein that was made. It goes through the Golgi, gets processed and modified, and then it comes out the trans side. And then it goes and does its job. The cell is always making proteins for a specific job. So these are different things that the Golgi can do. 3 things can happen. So pathway A, secretory vesicles can go out of the cell via exocytosis. That's one way. Vesicles that have lipids or transmembrane proteins, they can become part of the membrane, like maybe it's a channel protein. Or lysosomes, if it's a lysosome that was made, they have digestive enzymes, which are proteins. So maybe the cell's just making these vesicles, these lysosomes, and then it's going to sit there in the cell until it's ready to digest some. So those are your 3 things. It can leave the cell, it can become part of the membrane, or it can be used inside the cell. Okay, so here's the rough ER making a protein. The protein goes to the Golgi. Golgi processes it, and then either it leaves the cell, or the protein becomes part of the cell, or the vesicle stays in the cell for some purpose, like it becomes a lysosome. Okay, peroxisomes are special little sacs. They have detoxifying substances in there, so you can think of hydrogen peroxide. And it is a type of peroxide that's in them. So one thing that needs to be taken care of in a cell is free radicals. They are toxic, and so the peroxisomes can break them down so they don't harm the cell. And they have oxidase, which is an enzyme, and catalase, which is also another enzyme, converts hydrogen peroxide into water. So those are just going to help you clean up your cell and get rid of the toxic stuff. And then lysosomes are the digesters. So you make protein digestion, like digesters, like enzymes that are proteins in your ribosomes, and then they're going to be sacked up in your lysosomes and wait to digest things. So they'll digest, if it's like a white blood cell, it'll digest a pathogen like bacteria, virus, toxin. If you have an old organelle that you don't need anymore, they'll break those down. They help break down glycogen. So they just take care of any digestion that needs to happen in the cell. These are lysosomes, and then the green is where stuff is being digested. So they're just little sacs with chemicals in them that will digest stuff. Peroxisomes look silver. Okay, so there are diseases with every organelle. There's a disease. If you have something wrong with your organelle, there's a disease. So lysosomal storage disease is when the digestive enzymes that you're making are wrong. They're not functioning. They're mutated. They're not structured correctly, so they're not functioning correctly. Tay-Sachs disease is one example where they don't have that enzyme, so they can't break down glycolipids in brain cells, and it's a nervous system disease. Okay, so the endomembrane system is that pathway from making proteins in the ER, processing them in the Golgi, and then secreting them out in the vesicles and lysosomes. It's like that step-by-step process. So all of these organelles work together. They can produce, degrade, or store or export things, and then they can get rid of harmful stuff. So your endomembrane system starts in your ER. So the mRNA comes out of the nucleus, that messaging. You make proteins, then it goes to your Golgi. Your Golgi processes them, and then your vesicles kick them out of the cell, or you have a secretory, I don't know why I can't say that, vesicle, or you make a lysosome. So that is the endomembrane system, those steps from ER to Golgi to vesicles. Okay, then you have the cytoskeleton. So in your cytoplasm, you have like these structures, these protein structures that are kind of like the bones and muscle of the cell. So it's not just like a blobby jello. It actually has like structures inside the cell that help move it and move things around. There are little highways to move stuff around in the cell and all of that. So you have microfilaments, intermediate filaments, and microtubules. So microfilaments are the skinniest, the thinnest. They actually are what are in your muscle cells that help your muscles contract. So you have a cross-linked network of microfilaments, and it helps the cell not get squished. It helps it stay in the right shape. It helps it move. It also assists in endocytosis and exocytosis. So these are actin microfilaments. This is exactly what moves your muscles. And then intermediate filaments, kind of a medium size. They're tough, insoluble, rope-like. They help the cell keep its size, like its shape, help it resist pulling forces. They have a role in desmosomes too. So here is the picture, and you can see they're a little bit thicker, like a rope, right? And then finally, you have microtubules. These are the largest. They're like tubes, and these help the cell to divide. So they help move the DNA apart when the cell's dividing. They help move the organelles around the cell. They're just kind of the moving guys of the cell, right? And so here's what they look like, and you can see they're much bigger. So you've got all three levels of cytoskeleton in your cells. And then motor proteins are another type of cytoskeleton, and they also help. They're like they use the microtubules, but they help move stuff inside or out of the cell. So they're just a specialized type. I don't know if there's a picture, there's not. Okay. So microtubules can also form as centrioles, and those are the guys that help move the DNA. They separate the DNA during mitosis and meiosis. They also form cilia and flagella. So they're like a special version of the microtubules. So here they are. You might have seen pictures of this. They form during mitosis, but they also form other times to move things around the cell. So they just kind of congregate into that shape, and then they can do the work. And there's a side view of them. They're pretty cool. Okay, you have cellular extensions coming off your cells, including cilia and flagella and microvilli. So cilia are the small little hairs. Flagella are the long tails, and they help the cell move or help things move along the cell. And then microvilli are little projections like in your small intestine and large intestine, and they help absorb nutrients. So as the food material, the chyme passes through, they are absorbing nutrients because they're increasing the surface area. So cilia, whip-like projections, and then flagella are longer, like the tail on a sperm. Okay, and they have certain patterns. They're made of microtubules. So here is a cilium. There's the microtubules. So they're just, these microtubules are like multi-purpose, right? And then this is how they work. So sometimes they just move like mucus across them, or sometimes they actually help the cell itself move. And then you can watch that video. Microvilli are the little projections. They absorb nutrients and water, and they have really high surface area. So here's what they look like. And there's a disease called celiac disease. And what happens is your microvilli are shorter for some reason, and so they're not able to absorb as many nutrients. So that's a good example of structure and function. If your microvilli don't have the right structure, they're not able to do the right function. I skipped one, sorry, I don't know what it said. Okay, then you have your nucleus. Your nucleus has three parts: the nuclear envelope, which is a double membrane phospholipid bilayer that's around the nucleus, same as the cell membrane. You have a nucleolus in the middle, and it's shown as like a darkened spot, so it's like another circle. And then you have chromatin, which is the DNA. So here is your nucleus. This is the nucleolus. That's where ribosomes are made, rRNA for ribosomes. And then in the nucleus, that's where your DNA is stored, and that's in the form of chromatin. And so most of the time, your DNA is just like loose, relaxed, so it can be coded and used. And then when the cell gets ready to divide, it folds up into solid chromosomes. Not solid, but compact. So you have a double membrane. You also have a jelly-like fluid, like a cytoplasm, but it's nucleoplasm. And the outer layer is right up against the rough ER so that the mRNA can come right out and code for a protein. And then you have an inner layer too. And then you have those little orange holes, those little pores, and that's where the mRNA can get through. So here's your nucleus. You can see the zoom in of the little pores. And this is the nuclear lamina. So that you have little filaments in there too. So the nucleoli looks darker than the nucleus, and it's right in the middle. So if you've looked at a microscope, sometimes you can see them. In the nucleolus is where rRNA is made, and you usually have one, maybe two of the nucleoli. And then the chromatin is your DNA. It's also made of histones and RNA. Histones are proteins that help organize the DNA, and it organizes it onto structures called nucleosomes. That's when you have a few histones with DNA wrapped around it. And then chromatin is when the DNA is condensed, and it does that to cell division. So here's your DNA wrapped around on histones. So those are the little proteins. When you have 8 histones together, that's a nucleosome. And oops, oh, I skipped it, sorry. And then you zoom out and you can see the whole chromosome. All right.